From Microbe TV, this is Immune, episode number 82, recorded on July 23rd, 2024. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, everybody. How are you doing, Vincent?
Good to see you all again.
Yeah, good to see you.
You know, here in New York City, it's 28C and cloudy. Let's see what the humidity is.
I don't know what it is here, but it's
Yeah.
definitely cooled off. We had a really hot spell, massive wicked thunderstorms, and then since then it's been nice.
68% humidity. It's a bit humid today. Also joining us from Cleveland, Ohio, Steph Langle.
Hey, everyone. Great to see you. Good to be back.
And from Madison, New Jersey, Brianne Barker.
Hi, great to see everybody. It feels like it's been forever since I've seen y'all.
I know.
When did we do an immune with all of us? Do you remember? Was it
With
May?
all of us would have been April,
Yeah.
Wow,
Oh,
long
wow.
time.
Really?
I think. Pre-AAI. So we
yeah,
went to the
yeah.
American Association of Immunologists meeting.
Mm.
Would have been the month before that.
Well, here we are, and
Here
we
we
have
are.
some cool science for you today. But first, let me remind you, if you enjoy these science programs, we would love to have your financial support to continue them. Microbe TV is a nonprofit entity. It's a 501c3, so any donations you make are federal, U.S. tax deductible. You can go to microbe.tv/contribute. So today we have a snippet and a paper. And probably in the tradition of Microbe TV podcasts, they'll end up both being papers because they're so interesting. But first one is from Brianne.
Yes. So the paper that I chose is Human SARS-CoV-2 Challenge Uncovers Local and Systemic Immune Response Dynamics. This was published recently in Nature. Two co-first authors, Rick Lindeboom and Kaylee Warlock, and two co-senior authors, Marco Nikolic and Sarah Teichman. And these authors are from the Wellcome Trust or the Wellcome Sanger Institute in the UK. A lot of other authors, most of whom are from London as well. And this came from a study that I know we've talked about a little bit on TWIV. I don't know though if we've mentioned it on Immune, where a group in the UK did a human challenge study with SARS-CoV-2. So they took a small number of patients and infected them at a known time with a known dose of a known viral sequence of SARS-CoV-2. They had looked for comorbidities and done a full health check beforehand, and they kept these patients in quarantine. And there's certainly some controversy about whether that was a good or bad idea to have done the study. But what we are seeing here are immunologic data from 16 patients who were infected with SARS-CoV-2. And because of this challenge study, which, I mean, this is usually why we do animal experiments, because we can do infections at a known time and look at sort of all of these details. But here we have it in humans. They were able to actually see the early events following infection. They were able to see things like what might happen in someone who doesn't have symptoms yet or pre-symptomatic. They could look at people who were exposed and not infected and what kinds of immune responses they were making. And so I think that this is really fascinating data from this group of 16 patients, particularly using kind of systems immunology and getting huge amounts of data on what was going on with the immune responses in these patients.
And did you mention which
so
virus
the
they
patients
used?
were challenged. Oh, sorry.
Did you mention which virus they used? They used the original one, right?
Yeah, they used the original SARS-CoV-2 pre-alpha, and this was also done before most people had been infected and before people had been vaccinated. So this was the challenge study was done pretty early in the course of the pandemic.
Okay, because I was reading it's coming out now, and I said, well, where did they find people where they found them totally seronegative and had not been exposed? I was like, how did they do that? Then now I realize I didn't look at the details and the methods that they did this. In 2020? I guess they would have originally done this.
I think it was 2020 or 2021. I didn't go back. They
Okay.
have another paper really looking at sort of more virology
Mm-hmm.
outcomes in this cohort of individuals, and I didn't go back to look at that paper. I know that we have a TWIV episode talking about that paper. So just because of all of those things and the uniqueness, I was excited by this paper
Yeah.
and the things that we might learn from it. And so one of the things that I really liked was they were able to divide the individuals into 3 groups: those who had sustained infection, who had sort of multiple positives with SARS-CoV-2 PCR, sort of as they had expected infection to look like. A group who had what they referred to as transient infection, so who had some positives, but they didn't sort of qualify with the official definition that they had decided before the paper of infection, and those who had abortive infection, who were given the virus and who made an immune response, but who did not actually ever become positive. And that's one really nice thing that they can see in this kind of study that you can't normally determine. And what you can, of course, look at is what happened in immune responses in these 3 groups and what things helped make that abortive infection perhaps be abortive.
Right.
And just a question, like when we do studies in mice, we look for positive by whatever marker we're going by, and then we're using the ones that are not positive as uninfected or, for whatever reason, but we still look at immune responses. And this makes you think that, if you get this abortive infection and you have an actual immune response, but never a positive, how does that influence how we look at the mouse studies that we have been doing?
Yeah, I even think about this with some in vitro studies where cells are infected in a dish, and some of them are called uninfected but exposed, whereas they may or may not have been abortively infected and then controlled that infection really early. And I think that I am sometimes frustrated that distinction isn't brought forward. There are a couple of papers relevant to something that I think about in my lab that argue about this point, and I kind of argue that those 2 papers are actually just defining uninfected and abortive infected differently, and they say the same thing. And so that's something that I've always kind of wondered about, and I was really excited with this paper that they were actually talking about those patients as abortively infected patients, and letting us think about what types of immune responses might happen or might not happen when the viral infection is blocked from going through a full infectious cycle.
And they did not do a mock infected, so no virus, but same fluids and everything? Because couldn't that technically induce damage and cause some inflammatory response?
They did not do that here, and you're totally right. That could have.
That's why we always do with animal experiments a sham inoculations, because there's always some type of damage made, at the inoculation site.
Yep.
Okay. Well, things to think about.
Yeah. So, like I said, they used 16 patients, and they took samples from the nasopharynx. The patients had been intranasally infected, as well as PBMC blood samples during this. And while this isn't the first thing that they mentioned, to me the most surprising thing that I saw was that they found interferon responses in the blood before they found interferon responses in the nose.
Yeah.
Even
for the transient ones, right? Was it for all of them?
No, it was actually for sustained. So neither transient nor abortive made any interferon. Only sustained made interferon, and they made the interferon in the blood before in the nose.
And over baseline.
Yes, over
It could
baseline.
have some level of, right.
Yes.
They said that could be an artifact of sampling, right?
Right. They said that could be because they didn't sample, other parts of the nasal tract or something like that.
It's
Yeah.
weird that it would be in the blood first, isn't it?
But that was one of the first things that just really caught my eye because
is
I
it
found
a
that
sensitivity issue though? Because I mean, when you're doing nasopharyngeal swabs, maybe the level of interferon that you could detect by that is pretty low, whereas in the blood it's pretty clear.
Yeah,
Maybe.
that is interesting. So we deal with this. We're using ferrets pregnant lactating ferrets to infect with flu, and we do nasal washes, we do oral swabs. In the nasal washes, we always have a harder time finding the antibodies because it's a wash. We're diluting it, and if we wanted to look at the antibodies up there, we'd have to necropsy the animal. There's no real way, unless you do some type of scraping. So I agree. I think this is the crux of the issue.
The other question is when, I mean, I don't know if you guys got the original nasopharyngeal swabs when we all had to get tested for COVID.
Yeah,
I mean,
was
everybody was
time.
like, this thing is up in my brain, right? I mean, it was not pleasant. So I wonder what kind of damage that does also to the tissue and whether that might induce a little bit of inflammation in some of this too.
Yeah, that's a great point. So besides the interferon, they looked at immune infiltration into the nasopharynx, and they saw that in the sustained infection group and the transient infection group, they saw lots of immune cell types coming in. During the sustained infections, this really started about day 5 until day 10. In the transient infections, however, the immune infiltration started at day 1. And so it seems like one of the things that perhaps was involved in that infection being transient was an earlier infiltration of immune cells. And in the abortive infection group, they saw much less infiltration, but they did see some infiltration again on day 1. So that I think was a particularly interesting type of thing. They also looked at different types of antigen presenting cells, particularly different monocytes, and so they saw that there were changes in inflammatory monocytes in circulation, which they took to mean that the circulating monocytes had infiltrated. So that was sort of interesting. So they did look at some at the site of inoculation, but they also talk a lot about numbers of circulating inflammatory monocytes and whether they saw a decrease there. And it seemed like the monocyte changes were the same in all 3 groups. And so the early monocyte changes were the same in the abortive, transient, and sustained infection. And it was sort of other cell types that tended to be more different in the abortive infection group. They also looked at mate cells. They looked at gamma delta cells. And they also looked to see which cells were infected during this by looking at PCR for SARS-CoV-2 antigens. And they saw that they could get some positives from CD8 T cells, which was unexpected, some in epithelial cells, but that they saw really large numbers of infection, hyperinfection, which they said was more than 1,000 viral RNAs per cell on average in the ciliated cells, which they hadn't really expected. And
why didn't they expect that? Those are the ones that are at the barrier, right? Was there a reason why?
Yeah, and I remember there being even some papers early on about cilia having, I don't know if it was more receptor but they were a target for sure.
I think that in particular they weren't expecting such a high level of virus in the ciliated cells. And when they looked at the ciliated cells, it wasn't sort of an even distribution across the ciliated cells. It was sort of like they were small patches
Interesting.
but they couldn't find anything unique about those particular ciliated cells. And so I think maybe that part was more of the unexpected, as opposed to seeing any, they expect to see something in ciliated cells, but not a small number of ciliated cells having dramatically more virus than all the others.
Yeah, there's a paper of 2021, nasal ciliated cells are primary targets for replication.
Yeah.
But you would expect heterogeneity always, right?
Yeah.
Not every cell is going to be the same.
Yeah. I mean, they mention here that while the hyperinfected cells were 4% of all infected cells, they contain 67% of the viral RNA. So I think that maybe that heterogeneity, that level of heterogeneity was really the unexpected part. So they measured some additional responses in those ciliated cells to try to understand what was going on with the acute phase response. They also looked to see if there were any differences in MHC types between the three groups of patients. Given that they had 16 patients total, I think it's a little tricky to look at this, but they did think that having HLA DQA2 did seem to help protect patients from having a sustained infection. So that was sort of interesting. And then they started to actually try to find the T cells that were responding to SARS-CoV-2. And they did this by actually looking at a lot of different markers in the T cells to look at activated T cells and to measure T cell activation. And then went back and looked at the clonotypes of those T cells that were activated to see which clones were responding. And particularly, they wanted to know, were the same clones responding between the sustained, the transient, and the abortive infection groups. And it looked as though they really tended to see activated T cells in only those groups who had sustained infections, and they didn't tend to see a lot of activated T cells, particularly in the abortive infection group. And so it seemed as though the abortive infection group was controlling the infection before T cells were getting activated.
At least they haven't completely destroyed the textbook of immunology, innate immunity controls. And when that doesn't work anymore, then the adaptive immune system works.
But at the same time, when have you ever actually seen the data on that in a paper?
Right. Yeah, it's true. It's true. It's been the theory, but
right. It's sort of the idea. It's what I always teach, but
it.
now but here they're actually, showing it in detail in patients. They saw sort of the transient infection group was in the middle, where some of them had activated T cells, some did not.
And what was the timing of seeing those activated T cells?
Let me remember. So the peak was at about day 10. And then, interestingly, they started to see a peak of Tregs at day 14. To potentially, one could imagine, if one looked at the basic immunology textbook, that those might be starting to dampen
to calm
that inflammatory response. So they, looked through all of these T cells, and they then took all of this information and built a model that can be used by others who want to look at T cell responses. And so because they had done all of their single cell work, they have this huge description of what are the activated T cells at different days following this viral infection in abortive versus transient versus sustained infection. And so they made a searchable database that others who are looking at T cell responses in whatever model can now use and try to figure out what's going on. So I thought that was kind of cool and could be really useful for the field. They made also a reference atlas for a lot of data on COVID-19.
Right.
And they also have one other really important thing that I looked at, which was they tried to figure out how different types of responses happen temporally compared to symptoms. To think about what types of things do we see in a patient before the patient has symptoms versus after. Because you can imagine, of course, when we're doing diagnostics, we're usually diagnosing people with symptoms.
Yep.
And so trying to kind of figure out how all of this times with symptoms was important. And so basically, they saw that the earliest symptoms were happening about day 4. They were able to see basically their interferon change, the changes in mate cells, the changes in innate immune cells, all of those things before day 4. And all of those things before symptoms. And so the only thing that they looked at that was really sort of also happening around day 4 was a change in body temperature. They also did see that the loss of smell loss of sense of smell seemed to happen more around day 7.
Yeah.
I'm
I'm curious, did those, the watches or do they track your body temperature as well? I don't wear them, but like,
I think
No.
no.
These watches?
Yeah.
No.
Let me check.
I just was like, oh, if
Mine
that's
does
something,
not.
okay.
Mine only does at night.
It tracks your
Wait,
body temperature at
yours
night.
tracks body temperature at night?
Yeah,
Oh, wow.
I wonder why not during the day.
because I think at night it's assuming I'm not running around like getting sweaty.
Okay.
You're not? You don't run while you're asleep?
And are they thinking like body temperature is inaccurate when you're sweaty?
I guess so.
Yeah, there's no temp. Everything else is here. I mean,
What do you have? Do you have a what kind
Uh,
of watch do
Apple.
you have?
I have an Apple Watch. It is, I think is 9 an option.
Is it body temperature or is it ambient temperature in the air?
They list it as body temperature.
Oh, okay.
Really?
I've
Should be
heard it.
because it's got the contact of the metal with your
Right.
skin. You can
Yeah,
do
yeah.
you can do O2 saturation. You can do
Mine
your
has that.
Yeah.
Yeah.
pulse. You can do pulse easily, right?
I just, if somebody was very in tune with their body temperature and they wanted to know if they saw an increase before onset of other symptoms.
Right.
Mm.
That's
Well-
always the hard thing with treatment, right? Is it's very hard to treat for a viral infection early on.
Right.
Anecdotally,
I guess it's
yeah,
like
go ahead.
10.5.
Okay.
I guess.
Interesting. Yeah, anecdotally, a lot of the watches will give you some sort of like battery score, like where your health is or whatever, and it measures heart rate variability.
Mm-hmm.
And
Yeah.
I think heart rate variability is one of the very first things to change because people will know if they're getting sick because
Really?
they will
Okay.
see a change in their score for their heart rate variability
Wow.
or for their recovery after workouts and things.
Sure.
Also, I've heard, interesting for you, Steph, that people will figure out they're pregnant before they know because their heart rate variability will change
That is
significantly.
really interesting.
I don't know what that is. Maybe somebody
Sure.
who's listening knows, but I think that's kind of interesting.
I'll
Yeah,
have to
it's
keep
cool that
track
we
of that.
carry. Well, yeah, we keep track of so much stuff now just by what we wear. But that's cool. I'm gonna have to look up one for body temperature. I didn't know they could do
Yeah.
that. Interesting.
Most people can sense a little change in their body temperature.
I don't know. A little? Maybe.
Yeah, like a degree. I bet most people feel
Okay.
it. Yeah.
I don't know. I got 2 daycare kids, so it's a constant barrage of like sickness and viruses.
Yeah.
I never know what's
You're
going on.
either
I'm a mess.
symptomatic or transient or aborted.
Yeah.
For
I'm
everything,
never the
something at all
control
times.
group. Right.
There was another paper that I thought about choosing that came out recently that was saying that one of the reasons why kids might have had less symptomatic SARS-CoV-2 is because they had a higher kind of baseline interferon response.
I saw
all
that.
the time. Yeah.
I was on to that preprint when it came out. I think I forget Ellen Foxman.
Yeah, I think so.
Yeah. That's cool because and I know this is diverging. Sorry, but it kind of makes sense if kids are popped more with these pathogens. Could they have a higher levels of interferon?
And that makes it milder, right?
Right.
I thought it was interesting that these 7 people who were PCR negative still made innate responses.
I know.
Right.
Yeah,
It's very cool.
Yeah, it's very cool.
Well, and
No antibodies, right? But
no antibodies, no big T cell response,
Mm-mm.
but they still made responses. And I think that
Yeah.
we should always, just like Cindy was saying earlier, that's something we should remember in a lot of our studies.
And so the virus, it had to, it entered, it the innate immune system took care of it. So then,
hmm
but there's no viral, PCR couldn't detect it because it's not, I mean, it's sensitive, but it's
Yeah,
not
that's
sensitive enough.
that's weird, right? You would think. So that's how much you need to trigger innate versus PCR,
Yeah,
right?
yeah, right.
Right.
What's the difference there? Or the location issue again, right? Maybe we're
Could
just
be
not
location.
sampling.
Yeah. The other thing I wanted to just bring up about this, so they used a small number of people. They're
Mhm.
young, they're healthy,
Mhm.
but we know that young, healthy people get long COVID, right?
Right.
Yep.
Yeah.
So I don't know how you can do this ethically, knowing that some people might get long COVID and never recover, right?
So
'Cause-
there was a preview for this, right? I think Akiko wrote it
Mhm.
for this
Yeah.
paper. And I think she mentioned the long COVID thing. But again, coming back to my question at the beginning, where, how did they find these people that weren't exposed? Blah, blah, blah. If this was back in 2021, we would not necessarily have had a great handle on long COVID at that point. Like we knew about it, but it wasn't our everyday language. And so maybe they were able to
And I guess
do
the other
this
point,
before then.
I don't know if we're for sure that influenza doesn't cause long COVID.
Yeah,
It's
that's
just
I
been
bet it
around
does. Sure.
since we were born. So
I mean, I bet it does. Daniel
Yeah.
says everything, any respiratory virus causes
could
long
cause
something.
this.
Right.
Right.
It's just there's so many people infected now with this virus that it's
Well,
an
that's
issue. But
it's
I think going forward, you can't really do another challenge like this because you don't know. One person could get long COVID. That would be just wrong,
And,
right?
and
Yeah,
would you agree with
exactly.
then flu studies challenge? We shouldn't do those because that would be the same rationale.
It's true.
Yeah.
It's true.
Yeah, I don't know what I think about actually having done this study, but I think that the data, and I remember this from the previous paper as well, are fascinating.
Yeah, data are great. Totally.
Yeah, and
And you could think of
appreciate that they did as much as they could to make it publicly available. You
Yeah.
don't get a lot of
Yeah.
opportunities for this type of studies. So.
Yeah, it's not something that I've really seen in so many different ways, and I think it gives us useful information.
Yeah.
Just
I
not
think there's some fundamental stuff here,
Yeah.
right?
Yeah.
Too.
Absolutely.
That's good. I agree. And then you can think of all kinds of other experiments you'd love to do.
Yeah.
I mean, if we had a drug that would stop long COVID, then I would have no
Yeah.
problem with
Right,
that. But we
right.
Yeah.
don't.
Right.
We don't.
Great.
All right. Thank you, Brianne.
Yes.
Now it is time for Steph to talk
Wow.
about bats.
Sure. Well, I'm glad to hear the enthusiasm about this paper from the group. I think especially Vincent and Brianne, you guys seem to have an affinity.
You know,
Quote,
yesterday or the other day, I had seen this paper last week.
Yeah.
And yesterday I was typing an email to you guys saying we should do this. And
Oh,
then I
really?
saw yours saying
Oh.
I'm going to do this. So perfect.
Perfect.
Awesome. Oh, I do have very much an affinity for bat immunology.
Well, which is warranted because bats are very unique. So firstly, and I don't think I realized this, that bats are the second largest order of mammals after rodents. There's more than 1,400 species, which is incredibly diverse. And we know them as the only mammals, of course, capable of flying. And so to do that as a mammal, you have to have pretty unique physiological characteristics. And they do. So they have longer lifespans, low incidence of age-related diseases. And what's interesting for the TWIV crew, for the virology crew, is they do have a lower or higher tolerance to certain viral infections. They're chock full of viruses, in fact. They poop them out in their guano, but they don't seem to be affected. They rarely succumb to pathology or severe disease. And so that would mean that there's some evolutionary reason that they developed that to be the case. And other groups are looking into this. And so what I know of in the field is that how their interferon system responds, they seemingly can express interferons constitutively, which means that they might express higher levels of interferons always. Whereas ours, the way we described it is that they are expressed. You have interferon responsive genes and then it's expressed, but they may express these genes more. And then altered kinetics of these responses. And that also relates to the damage that happens to the cells. So damage-associated molecular patterns or DAMPs, cytosolic DNA, for example. Maybe they have impairments in the ability to detect them. So then you're not responding to those things, which causes the inflammation, which causes the immunopathology.
Yeah. From my reading, a lot of this goes back to that being the only mammal capable of flight thing. and how that requires different metabolism,
Right.
and that
Right.
metabolism causing more oxidative damage to the DNA,
Right.
and thus there's damaged DNA to be sensed all the time.
Right.
And so that kind of changes what the sensing looks like and what the basal levels of interferon look like.
Right. Right. So those are a few, to name a few, of the things that people are currently studying. What this paper wanted to study was antibodies. And so before I go any further, let me say the title of the paper and the authors. So the title is Temperature Sensitivity of Bat Antibodies Links Metabolic State of Bats with Antigen Recognition Diversity. And the first author is Nia Toshkova, and the last author is Jordan Dimitrov. Where are they from? Let's remember. I think
Bulgaria.
Bulgaria. Okay, thank you. I was like, I couldn't remember. Yes, Bulgaria. This is in Nature Communications. And they wanted to understand if antibodies were unique in any way in relationship to these different metabolic states. And the metabolic states that they're interested in, or the differences that they're looking at, are temperature. So they discuss how body temperature in bats can fluctuate, so that seasonally, but also just day to day. And that's because in their ability to fly, the metabolically demanding flight is usually around 40 degrees Celsius. And I wrote down what that is in Fahrenheit for the Celsius Challenge audience members like myself. 40 degrees Celsius is 104.
That's some pretty hot bats.
Pretty hot, right? But then they can drop to 20 degrees Celsius, which is 68 Fahrenheit,
Nuts.
during shallow torpor, which, I don't know, that wasn't a GRE word for me when I took the GRE.
Did you look it up?
Resting.
Resting. Thank you, Brianne. Thank you.
Yeah.
Yes. Resting. So inactivity is torpor. So when they're not active, which could be, I guess they're just hanging it out in a cave
and sleeping.
They're upside down, sleeping.
Torpor, the state of most humans, right?
Right. Yes. In modern day, yes. Or they could drop below 10 degrees C, which is below 50 Fahrenheit, and that's during hibernation. So, excuse me, you have both high and low body temperatures, and these are proposed as to be mechanisms by why infection could be different in these animals. So if you have lower body temperatures, viruses don't really like to replicate in lower body temperatures, or higher temperatures. There's this theory that mammals have fevers because your body temperature elevates to neutralize the ability of the virus to replicate. So both extremes, you can imagine this could be helpful for the organism. So this temperature, they wanted to understand if that changed the ability of IgG. So they're just looking at IgG antibodies. What we know is that antibodies come in many flavors. IgG is the most common in blood. It's the most common antibody measured in different studies because of ease of access to blood. So keep that in mind. But it can exist at mucosal sites. But what dominates the mucosal sites is IgA, which is a polymeric molecule, or IgM, which IgM in blood is a monomeric, it's one, but in mucus, it's a pentamer, there's 5, which is really cool. And then IgE, which is associated with allergy and parasitic infections. So we're going to just focus on IgG, whether or not they have, because this all is dependent on, do you have the reagents commercially available to isolate IgG from bats? And they seem to have figured that out. Other antibodies, I'm sure it's harder. So they looked at IgG in 2 bat species. And this is the greater mouse-eared bat, or Myotis, or the common noctule, which is Nyctalis noctula. You can correct me if you think that's incorrect, but they also looked at a couple different bat species, which we'll talk about, to confirm their results. But those are the 2 main ones. And so they went to, they purified IgG from the pooled blood serum from these animals, and they did an ELISA. An ELISA is an assay where you can coat the bottom of the plate with any antigen that you wish, and you are going to measure the concentration of antibodies that bind that antigen that's on the plate. It doesn't tell us anything about binding strength, which we will talk about in a couple of figures. This is just how much of it is there, and under these conditions, binding to a flat plastic piece of, plastic plate. And what they found is that when you expose these antibodies to higher temperatures, so which is the 42 degrees Celsius or 104 Fahrenheit, you have a greater amount of antibodies from that sample that bind to that antigen. That seems to be across from the multiple antigens they measured. I could name a few. They looked at, I think, are factors 8 and 9, are those complement?
No,
Or
they're
coagulation?
Clotting.
cascade.
Thank you. Clotting cascade, insulin, ACE2, hemoglobin. But then they get into things that are a part of pathogens. So Ebola, Nipah, Hendra, glycoproteins, HIV. They honed in on a couple that these bat IgGs bound very robustly to. And my interpretation is that these animals, that could mean that they have B cells that are specific to those pathogens. They were exposed to those pathogens. That's my interpretation. At least, for example, SARS-CoV-2 spike protein, they don't have many antibodies that bind to that. Although, like the others, with increased temperature, you find more antibodies that can bind. That is not the case for the humans. So they didn't see this phenomenon with the humans. They also didn't see this with other mammals they tested. I believe cattle,
there was a bunch.
goats, mice, and they also looked at chickens, which make a unique antibody called IgY, and this seems to not be the case for those animals. So it seems to be specific to these two bat species that they purified IgG from. They measured this based on the average standard deviation from the mean of all of those binding concentrations, and bats are winning in the ability to bind at higher temperatures.
And which I think is super cool. Somebody might sort of say, well, why don't they have SARS-CoV-2 or any other, is there a cross-reactive coronavirus? But the two bat species that they're using here are sort of insectivorous bats that are not the kind of bat species that we think of as the sarbecovirus hosts.
Nice. That's a good point.
Mm-hmm.
Great point. Again, speaking to the diversity of bats.
Those are rhinolophus bats,
Yeah.
right?
So,
Steph, this
yes.
idea of temperature sensitive, did this come out of the blue, or this is a thing in the field?
So there have been studies looking at polyreactivity of antibodies against different antigens, and they do find, and they do cite this in the paper, that it has been shown in humans and mice that the ability of an antibody, or the Fab region, which is the region that binds to bind an antigen, does increase with temperature. It is a little different, though, and this is the nuance. That is polyreactivity. And what they're going to describe is that even a monoclonal antibody will increase its ability to bind. So they're using polyclonal serum here, which means that all the antibodies are to different things. But in that polyclonal serum, you could have monoclonal antibodies, which have been affinity matured in a germinal center to bind one thing. Or you could have polyclonal antibodies, which is like if we want to get specific, T cell independent immune responses, and they are more broadly able to bind things. So it has been seen in humans, but I think what they're honing in on here is this ability of purified IgG to increase binding with increased temperature. So hopefully that makes sense. We'll get into it a little bit more.
Yeah, because go ahead.
I was just gonna say, if you look here, they really don't see it in humans.
Right.
It's
right.
And so in a lot of ways, it's not necessarily something that we're seeing in the species we often look at.
Right.
Yeah.
Yes. Yeah.
What I was thinking about is, do bats just have more polyreactive antibodies? Right? Because then they would be more likely to have this property. Whereas in the humans, they have more monoclonal matured antibodies, so they're less likely to have this kind of phenomenon. And is that responsible for the difference? And then I started thinking about, might temperature influence B cell maturation and B cell function, which I don't think they really addressed in this, other than just taking one antibody and showing that one antibody could bind to multiple things at higher temperature. So it is a property of the antibody, but it might be a property of the antibody and the host, right?
Right, which I think, and kind of will be a little bit of a, I'm going to give away my critique because I think this is important to discuss the nuance here. So you're right. This is not describing, in my opinion, the ability of the bat to diversify its antibody repertoire, which is in the title and is talked about a lot, that this is a difference in antibody diversity. I think this is a difference in antibody breadth. Because diversity, the ability to diversify your antibody repertoire comes from the B cell, and temperature could impact the B cell's ability to do that for sure. But this is taking an antibody that's already popped out of a B cell. Any affinity maturation has already happened. And now you're playing with the structure of the sequence of that antigen binding region. But the sequence stays the same. There's no diversification after you pull it out of the animal. So I think the title, I have antigen recognition diversity. It should be breadth. I'm just being a little picky. What do we think about that?
I'll leave it up to the antibody expert.
I will agree with you. I've been sitting here thinking about how, if there is a way you could actually try to make the B cell development happen at different temperatures, and I can't come up with it.
Well, I think you could isolate the B cells from the bats, sequence the B cell receptors, make monoclonals from those. You have to sequence the BCR to say anything about antigen recognition diversity.
Well, so you said something that they have already gone through, like somatic hypermutation and affinity maturation.
A proportion
that's
of them
an assumption, right? But do they know that a similar proportion of them have done that in bat and human? That's sort of my question,
there
right?
There was a talk on that at ASV.
Interesting. What did you glean from this talk?
My recollection was that it was similar, maybe slightly higher, but relatively similar.
Okay. So because my thought was, if the higher temperature somehow affects B cell interaction with, FDCs or their affinity maturation process or any of their gene, somatic hypermutation processes, if temperature affected that, you might have different maturity levels for the antibodies that arise in a bat versus those that arise in a human. And that could just generally, those antibodies will have this property just because they are more polyreactive because they're less matured on an antigen.
But I think to test that, you have to work on the B cells.
Yeah.
Because what they're doing with the antibodies does not change diversity. So
and everything they measured, you couldn't tease this out. But I don't know if there's a potential contribution of that also to what they observed.
If I had to guess, I would say yes. Because why not, right? Why wouldn't the B cell also be affected by this temperature situation? So I think that's something to remember, that generating diversity is something at the gene level in the B cell. Breadth is an outcome of that, but it seemingly can change based on things like post-translational modifications, glycans, right? That can change it. Or seemingly maybe change temperature. So you can really nerd out on some of these definitions, but so figure one is telling us that. Now with an ELISA, we just know that the amount of antibodies in your sample are binding more of the antigen with higher temperature. They want to prove that the strength of binding is improved, and they do that by something called surface plasmon resonance, or SPR. We like is a shorter, easier way. And basically what this is you're flowing your antigen over, you have your antigen immobilized to a chip and you're flowing the antibodies over top of it. And so there's a polarized light source that can detect the chip at various angles through like a prism. And the angle is known as the resonance angle, and the light does fancy things with the electrons. And so that light intensity that's reflected is how they measure this. And they have things called association phases or dissociation phases. And association is how quickly can the sensor sense that the antibody binds to the antigen. And dissociation is after you stop flowing it, does the sensor, how quickly do you see a decrease in response because the analyte is now dissociating because you stopped flowing it over. So what they show is that at higher temperatures, you have more antibodies binding and more strongly. In fact, they couldn't even calculate a dissociation constant in any of the temperatures until they did the high temperature because they just didn't see strong binding. And so that's interesting because when I think of the ability to expand breadth of an antibody, it's often associated with a decrease in binding strength. So to develop a broadly neutralizing antibody to HIV, which is a big goal, you want an antibody that can bind multiple different strains, but sometimes the strength of binding is lowered. Bats seem to be unique that they're not only increasing the amount of things they can bind to, but the strength as well. And how they do that, I think, is, of course, a future paper. But so strength is in both of those cases. They also said, okay, what about, this was purified IgG, what about whole sera? Because you can imagine there are other proteins in there that could be affected by temperature. They seem to see similar effects. They also start to then ask questions about, in looking at this first figure, the ability to bind to autoreactive antigens, and they use this cell called HEP2 cells. And I've not worked with them, but looking into it, they thought they were a laryngeal cancer cell, but they actually were just HeLa cells contaminated into this laryngeal cell line. So they're HeLa cells essentially, but they express a lot of autoantigens in their nucleus and their cytoplasm, and clinically you can diagnose diseases like lupus.
Yeah.
This is one of the ways, if they bind to these cells, it means you have a lot of autoantibodies. And it seems that the purified IgG from the bats only bind these cells at higher temperatures, but not humans. This is not the case for the human IgG. Okay.
And
so
you can tell based on the pattern of staining whether people have anti-DNA antibodies, anti-RNA antibodies, or this somewhat generally. If people are good at reading those slides, they can tell you what kind of autoantibodies someone has in their sera,
right,
when they do that staining.
Yeah, I feel like people who are kind of antibody biochemists very much want the sequence of these antibodies right now to look at what might be going on here.
Totally. I think that you're right. That is the next step now. I'm going to explain some more cool tricks that they do to look at the structure and function. But I think now it's like, okay, what is so unique about the sequence that gives it these functions? The next thing they want to ask is, okay, well, we've tried a series of antigens. There's like maybe 15 or 20. We're going to try 17,000 of them.
Yeah.
A variety of them. And this is a microarray. This is a company makes this, and they put all these antigens on a chip or a plate, and you add your sample, and then you can look at the ability of the antibodies to bind, and they see that bats are just able to bind way more. When if I could find that number, I think it was like a 10-fold difference in the number of antigens that bat antibodies could bind to, particularly at higher temperatures.
Right.
So it's not specific to any one thing, which I think is leading down this road to these antibodies maybe being more polyreactive to different antigens.
Yeah.
Okay, so then they wanted to dissociate the ability of the FAB region to bind, which is the part that does bind to the antigen, compared to the FC region, or the constant region, which has its own special functions. Those can anchor and bind to FC receptors on innate immune cells and flag for opsonization, complement deposition, multiple different things. So I think their first thing was, okay, if we cut off the FAB region, we cleave it off with an enzyme, do you still see the same effect of just the FAB2, two FAB regions? And it seems like they do. They tried 6 different antigens, and you can see that the binding increases with temperature. I will say one experiment that would have been good here is we don't actually, okay, so we're going to talk about FC dependent interactions in a bit, but to really say that the FC region doesn't contribute at all to this effect, I think you have to take the same sample, run it like they did, cleave it, and do the same assay compared to the FC only, because FC can stabilize FAB to particular antigens. I think that would have been a nice experiment to compare and contrast to if we're really saying FC does nothing here. Picky. Maybe I would have asked for it as a reviewer. I don't know.
And coming back to sort of this geeking out thing, avidity versus affinity, the fact that you have the two binding sites, and there is sometimes this, if you bind one site, the other site binds better. And so you have this avidity increase. They didn't, as far as I could tell, look at that at all. And, one of the things it could have done would be just use the one binding site, right? And see whether that had a difference in binding to the antigen with increased temperature, or perhaps it's some sort of flexibility of the antibody at the hinge that allows avidity to increase with temperature. Therefore, you get more binding.
That's a really good point. And there are assays where you can add particular chemicals that it is made to, once you bind it, to release the antibody, and it's in a measure of avidity. I think that's an assay they could have done to address that, because I agree.
They could have also thought about ways to make fabs instead of fab2s.
Exactly.
Right. To your point, yes, because that increases the avidity just by having 2 over 1.
Correct. Yeah.
Great point. So they wanted to ask, does FC binding to its receptors have the same effect? And they picked 3. They picked C1q, which binds to the FC region in the complement cascade. They chose FcRn, which stands for neonatal FC receptor, but is not just expressed in neonates. It's meant to recycle our IgG back into our blood, or bring it for degradation. Or if you're the placenta, it just crosses right on through. It doesn't recycle. So FcRn can bind. And protein G, which we use to purify IgG, it's a bacterial produced protein, I think. You can buy it commercially and pull out IgG from serum. And they did this same type of experiments with the higher and lower temperatures. They only did here 22 degrees and 40 degrees, and they actually saw a decrease in binding of these different proteins to the FC with higher temperatures. And that was in all 3 species, which was, 2 bat species and humans. So maybe just degradation of the FC is what caused it, but it was not a bat-specific phenomenon. This was across the board. Okay, so that then led them to, we know that this is a fab region phenomenon. So we'd like to know a little bit more about what are the characteristics of the antibody that are making this true. And so I think now I am on, where are we at here? Figure 4.
Yeah.
And they did a couple different things. So first thing they did is looked at thermodynamic stability. And they did this using something called a thermal shift assay. And it's based on the principle that a protein will unfold when it's heated, it will denature. And so the temperature that a protein denatures, that's called the melting temperature. And here they use a fluorescent dye that will bind to the hydrophobic region of the protein that becomes exposed when it unfolds, so as it denatures. So the more denaturation, the more the dye can bind in fluorescent, that's the signal. And it seems that the unfolding temp, or the melting point of the bat IgG, is lower than that of humans. So that indicates a lower stability. So it does not take as much heat to break, to kind of burst open these protein interactions, which could explain maybe some of the things they're seeing. They then wanted to look at hydrophobicity, so the hydrophobic interactions between the molecules and those hydrophobic groups that are attached. So they use something called hydrophobic interaction chromatography, and this is a type of liquid chromatography that you can separate and purify proteins based on hydrophobicity. And they use the hydrophobic groups attached to a resin, and it's that interaction that they're reporting here, and they don't see any differences. Now, this is interesting because in about 2 more graphs, we're going to talk about some in silico evidence that the hydrophobicity actually is increased in bat IgG, but only in a very specific part of the Fab region. Here, they're just using they haven't cleaved anything. This is just bulk IgG. So my hypothesis is that if they would have done this by cleaving the Fc off, maybe they would have seen a difference. Okay, so next thing is, well, let's look at, and they actually, I should say, to give them credit, they did mention that. They said that the sequence similarity between bat and human Fc fragments is why maybe it didn't work for bulk. And I would have said, oh, okay, well, just go ahead and cleave that off and try it again. See if you get differences. But the next thing they wanted to look at is ionic strength. And so ionic strength broadly is the measure of ions in a solution. And so it affects electrostatic interactions between charged molecules. Everything has a charge. And so antibodies and antigens are not different. And so a higher ionic strength can shield electrostatic charges, and it can change the attraction and repulsive forces. So what they use to measure this is you add an increase in amounts of sodium chloride to a solution, and in water, NaCl will completely dissociate into positive and negative ion. And so you can control the strength. You can have lower ionic strength with lower amounts of NaCl or higher amounts of NaCl. And that's what they used here. So interestingly, they just saw different effects with bat and human. It wasn't exactly clear what those effects meant. So for example, in the bats, they said that they have this V-like formation where you have low NaCl, you have high binding. You increase the NaCl, you have lower binding. You keep increasing NaCl, and you again have higher binding. So it seems like bat antibodies are flexible to changes in electrostatic forces at both high and low ionic forces. I don't know. Did you guys? I'm like,
yeah, I don't know,
okay,
because for the
cool.
human antibodies, they stay pretty stable. For you keep increasing the NaCl, it's pretty stable, and then it starts to drop off when you get too high,
right?
Which makes sense. So I don't know. Maybe there's some protein biochemists that can say, oh, that's what that means, but
Yeah.
I have no idea what that means. It's just like, interesting.
Yeah.
Well, and I also couldn't figure out if I cared that when the bats if you look at the bat antibodies, when they dissociate, they're actually going much lower on
That's a good
the
point.
y-axis than are the human antibodies. So if you look at the bottom of the V, the bottom of the V is much lower than you see at any point in the human antibodies. And I don't know how to interpret that.
Right.
And to know whether that's something that is interesting or important.
Other than I think maybe what the point that it enforces is that they just have this flexibility to extreme conditions, and whatever the sequence, the protein structure is very it can react to different states. I guess my other question is, in a bat, like what is the normal
Salinity?
ionic state?
No idea.
I don't know.
That would have been helpful, maybe.
They admit that they don't know what any of that means.
Oh, okay.
Yeah.
I mean, they say these properties could be associated with the binding properties, but they don't know how. Right?
Yeah.
Right.
It's almost as if someone made them do these experiments,
Yeah. Yeah. Like what are the forces?
Well, I mean, a prediction would be that there must be something different about the hydrophobicity or the electrostatic interactions between the antibody and the antigen that might account for this. Right. That would be the hypothesis, because those are the two main forces that drive antigen antibody interaction. Right. And basically what this is saying is, yes, the hydrophobicity and the electrostatic characteristics of these antibodies are in fact different from what we commonly understand about other antibodies.
Right.
So so it could have some contribution.
Right. And I think what we can take away from it is just how different that it has this like a biphasic response.
Yeah.
So that was neat. Then they looked at in silico work, which is taking the sequence of the heavy chain variable regions of both bat and humans and using their program that I'm not familiar with at all, but to predict hydrophobicity. And it seems like when comparing the sequences, now they couldn't use their bat species where they originally were working with, which is the M. myotis. They had to use Myotis lucifugus. Somebody can correct me, because their genome is more sequenced, it has better annotation, and so higher levels of hydrophobicity of that particular sequence, which is an important sequence in humans as well, because it's where a lot of diversity happens. It's where a lot of important binding interactions happen. So it's relevant to the story. Okay, so the next thing they wanted to ask was this idea of polyreactivity. So polyclonal, right, again, is just that you have a pool of antibodies that are all targeting different things, but something that's polyreactive means that one antibody can bind multiple things. And so there is a monoclonal that you can buy called ED38 that's a human antibody that has this property, whatever is unique about it, and they're showing you it can bind multiple things. And as shown in the past, this polyreactive IgG can increase its binding strength with higher temperatures. So while the bulk IgG from a polyclonal pool did not show the same effect in bats, one monoclonal that is polyreactive does. So that's, I think, the nuance there. But that is not true of other monoreactive monoclonals like VRC01, which targets an HIV glycoprotein, or S309, which I think is a SARS-CoV-2 spike. I don't remember. I think SARS-CoV-2. And they didn't see temperature differences between those. They then asked questions about binding strengths in comparison to those antibodies, bats compared to the monoclonals. And it seems like bats have higher of their polyclonal pool, more of them are polyreactive. And that's important here because it is an ability of antibodies to, even though their B cells may be reacting to one thing, that under these different conditions, the antibody shape or charge or whatever can change to bind many things.
So is that a good thing or a bad thing?
So they describe that, and in these couple next experiments, that if you want to bind up a lot of dead cells that you're producing in your high metabolic state for optimization so that you can degrade them and get them out of your body, it's good. But I was thinking, we don't want to make monoclonals for therapies out of bat antibodies for humans, right?
Mm-mm.
Because they could target many things.
Mm-hmm.
And that is what that's what autoimmune disease is.
Right.
Right.
And this goes back to the points earlier about sort of B cells and B cell development, because this makes you want to know, well, what's happening in terms of the central tolerance mechanisms in these B cells, and are they different? Are the same ones active? Are some of those central tolerance mechanisms not happening? Like what's up?
Yeah.
Oh, well, that makes me think about, like, what about thymic development and selection and, just in general, the antigen development of, anti-antigen responses in general in the bat. Could that be affected by, from the time the bat is very young, how those work? So how are autoantigens presented to do selection of T cells?
Right. And is that relaxed
and then,
a bit?
Yeah. So are these, do we know, are they T dependent, or are these in the bat mostly T independent? And could that have something to do with it? Is it more T independent antibody responses develop with higher temperature, or are there ways to think about some of these things? Because I'm trying to think, how does this apply to what we know in human, besides what you just said, which is we don't want to develop bat antibodies to use in human immunotherapies because they might just be broadly reactive, especially if you get fever and then you get autoimmunity, and that would be bad.
Right. Well, and I guess the other question, and this is sort of a silly question, is how do we measure autoimmunity in bats? And are these bats flying around with
Yep.
like arthritis.
Yeah.
Well, they hang upside down, so maybe they wouldn't notice because their joints are always stretched and not pressed by gravity, right? Maybe
That's
that
interesting,
has
yeah.
less of an effect.
Yeah,
Yeah,
they're
like-
not really walking or creating the tension on their joints.
Yeah,
But
so
presumably flying would be difficult if you had pain.
Yeah.
But these are long-lived animals,
Mhm.
right?
Right,
That's the
right.
other thing. These are not like short-lived, and if they get a little arthritis, you're going to weed out those. These are relatively long-lived animals. One would think that if they had issues like that, it would,
but how would we know?
I don't know.
It's hard to believe that a species would live with autoimmune disease. You know?
Right.
Right.
But they
I
seem
know,
to
but we
they
don't
seem
know.
to suggest that it's endothelial and epithelial
Right.
cells that have this. So
So they use endothelial lysates, and they also use autologous epithelial cells of the bats themselves, and they see that the bat antibodies can bind to the components of the epithelial cells. To human, I think they used foreskin fibroblasts or epithelial cells, something, to show that, yeah, they can bind to all kinds of proteins in cells that are naturally in their bodies.
So I have one little
Hmm.
issue with this experiment, and I went and looked it up because I was really curious about the timing of this. Because so what they're doing is they have live cells,
Yeah.
and they stain for annexin-5, which will bind to phosphatidylserine on the surface of apoptosing cells.
Right.
When they lose their ability to maintain the membrane, this phosphatidylserine flips and it's exposed to the outside world, and macrophages can use that to eat them, but you can use the stain to see it. So you can see apoptotic cells. Propidium iodide is a non-membrane permeable dye that once the membrane is compromised, then propidium iodide can get inside, and that tells you necrotic cells. And then if cells stain with both, it means they were going through apoptosis and then they kind of went through secondary necrosis and just kind of. So they did this study with hydrogen peroxide exposure.
Right.
And the flow cytometry plot that they shows is basically 100%-ish, 92.3% of the cells are alive, so PI negative, annexin-5 negative in the live condition. And then in the dead condition, it's basically almost 80-90% are double positive. So it's pretty much the whole population is now secondary necrotic. So there really isn't any apoptotic cells, which you presume in the bat, if there's going to be endothelial, epithelial damage, whatever, you're going to detect it early and eliminate those cells. You're not going to wait until they're
all of them are dead.
fully dead and like fallen apart into bits and pieces and completely destroyed to be detected. But they didn't really look at any individual in anything in between. And when cells are that compromised, the proteins are going to start to unfold. Weird things are going to get chewed up. Bits are going to be exposed that are not normally exposed. So you might have hydrophobic patches everywhere. And based on the changes in the antibodies, maybe it just binds because of that. And it's not anything other than there's just more sticky crap if you completely ablate the entire cell, right? So it's a little, I think it could be better done if they're trying to make the argument that if in the bat with a lot of high temperature and more reactive oxygen species generated, you have some cell damage, and when the cells are damaged, you need to detect them and eliminate them. That's a little different than what they're detecting here, because this is really way far past those initial responses where you'd want to pick up those cells.
Yeah, I think a dose response curve or some type of dose response would have been great, because you're right that they show that, but I think their argument be, well, it's only at the high temperatures and only in bat that you see the binding. So
True.
Even though, it probably is a lot of dead sticky stuff, it is specific to bat. But so biological relevance then would matter. Okay, you show it in the extreme setting, but okay, now let's back off the hydrogen peroxide in these intermediate states. Does it matter?
Or just treat with something that induces apoptosis, and can they eliminate those? Can they recognize those apoptotic cells?
Right.
Or
does
use
this
something
only-
other than annexin for the measurement.
Yeah, true. Yep.
Yes. And so that, kind of summarizes the final two experiments, as they showed this using human cells, that, endothelial cells, and also using bat epithelial cells.
And that last Experiment? I mean, I don't know. They still recognize live cells and they
Mm.
still recognize it at cooler temperature. It's just less.
It's just less. So yeah, I think it's not I think they're arguing it's not that these antibodies don't recognize anything at torpor temperatures. It's just that for whatever reason, they have a better ability to bind these at higher. And so maybe the next experiment is that, okay, right, does that matter for flight? Is that why it was evolution drove this process? So I think that summarizes the paper pretty well. Bat antibodies are cool. They
Actually, they're hot.
They're hot.
There's our
That's our
title.
Yeah, I love it. And it seems to be something specific with the sequence, particularly of the heavy chain variable domains, which we know are special in antibodies. And so future work, I'm excited to see what this group finds.
I like
Do you
the. Oh, go ahead.
Go ahead.
Oh, I was just going to say, I think there was an interesting thing in the discussion where they suggested that maybe this has something to do with when they come out of torpor, or hibernation. So when they're at very low temperatures, like you said way back at the beginning of this, is that the virus's replication is not very good. But as soon as you raise the body temperature up, the viruses could start to grow and replicate, and therefore changing these antibody properties very quickly to be able to recognize the viruses quickly might be a evolutionarily developed thing to deal with this unique aspect of these bats. I thought that was kind of interesting.
Is there any way to test this idea that this temperature sensitive property is essential? Right, because otherwise you're kind of speculating, making associations.
Yeah. Could you infuse antibodies from human? Well, there's a lot of problems with that. I was just thinking, giving the bats antibodies that can't do this, like finding a way to prevent them from doing this and seeing if they can fly as easily, or but it would still be a transient effect, right? Because
What if you made a transgenic bat so you made them, they can make only one kind of antibody or whatever, and then see if that changes anything.
Vincent, Brianne, do any of the people you know who do this work, are there transgenic bats? No? Okay.
I don't think so. No.
Is there talk of that? Is that possible?
It's a good question. The bats in this paper are all captured, so.
Yeah, I mean, there are relatively few places where people have like a bat house.
So that's the experiment. Those animals don't fly like these guys. So you compare the serum from colony raised bats to wild bats, because colony bats, they're just not. And if you breed them for that, I just wonder if those animals can't do this as well.
I bet the colony raised bats would still have thermal properties of their antibodies, right?
Yeah, that's true. Because it's something that, yeah.
I think you could, well, in theory, it would be great if you could replace the antibody locus with like a human antibody locus. And then that should humanize bats.
Step one,
or
be able to grow bats in a colony. Step two,
how about
make transgenics.
Put the bat genes into a mouse.
And fly them? No, just kidding.
Well,
no, I know what you mean, though. Yeah.
I think the antibodies have the inherent property of being temperature sensitive.
And so then could you see the same effect in the mouse?
Yeah, so you put the genes into, I don't know how many you'd have to put in.
Well,
right?
yeah, that would be the question is it the locus or is it the central tolerance mechanisms?
Right.
Both, right, yeah.
Right.
But at some point, people have to test this otherwise.
Right.
So it's always speculation, but it's very cool. Thank you, Steph.
You're welcome. Yep.
It's I thought you guys would love this paper, yeah.
It's a winner.
And you did too. All right, that is immune number 82. You can send questions or comments to immune@microbe.tv. In fact, you can suggest papers for us. That works well for many of our podcasts. And if you enjoy this work, consider supporting us. Go to microbe.tv/contribute. Cindy Leifer is at Cornell University, at Cindy Leifer on X. Thank you, Cindy.
Thank you.
Steph Langle is at Case Western Reserve University, at Stephanie Langle on X. Thanks, Steph.
Thank you.
And Brian Barker's at Drew University, at BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.